run a mass spectrometry facility. I help researchers on campus achieve their scientific goals. They come to me for help with specific proteomics questions. I guide them through the whole proteomics workflow, basically. I help them set up protocols. I prepare samples with them. I show them the mass spec. I show them the data analysis, and I help them interpret the data. My mass spectrometer is a timsTOF HT. I have that coupled to either an NanoElute 2 or an EvoSep Eno. The projects that I use it for are very diverse. I kind of work on everything chemoproteomics related, but also non-chemoproteomics related. Mostly bottom-up proteomics, tissue proteomics, high-throughput proteomics, post-translational modifications, some immunopeptidomics work. We kind of try to do everything that we can on this instrument.
Yeah, when I started my lab, I bought a timsTOF Pro 2. This was in 2021. And I think this has been really transformative for my lab. When I started, a lot of groups would run maybe very small batches of samples, and they would be very, precious and almost like rarefied experiments where you would, invest tens of thousands of dollars into a single data set. But that data set would only encompass a few samples. And so the Pro 2, when it was introduced and came into my lab, we were able to run hundreds of samples in a given experiment. And we could do this at a very low internal cost. We could run this at maybe $5 to $10 per sample instead of thousands of dollars per sample. And, starting a new lab, this was really important for me because as a young assistant professor, you want to save money. But it was actually even better for my grad students who could then design new experiments fearlessly. They wouldn't have to come to me and say, this is a really cool idea, but it could be really expensive. That would be a big barrier for them to go do an experiment because they knew that it would be so easy to try a new idea. Oftentimes they will just do it and not even have to ask me. And I think that's the best thing. I want people to be able to have new ideas and just try them and explore things where failure is an option.
We recently found that in human tissues, there are thousands of abundant and stable proteins whose sequences are not predicted by the genetic code. Mm-hmm. And these are primary amino acid sequences that illustrates how little we know about the proteome, but also illustrates the power of mass spectrometry to start filling in these gaps, to start better characterizing our proteomes. And I'm very excited about the potential of timsOmni to contribute to this process, as it can provide direct evidence via hybrid fragmentation, different types of electron-enabled peptide fragmentation, protein fragmentation, to provide much more direct evidence so that we can start characterizing these new unknown proteins with much higher confidence, and not only discovering them, but also linking them to biological functions. And what is tremendously exciting and different about this moment is that we are finally at a stage where we can dramatically accelerate the rate of understanding proteins. Proteins are tremendously complex in dynamics. They're more challenging to study many other elements of our bodies, like DNA. There is a reason why DNA was sequenced first. The structures of DNA were characterized long before proteins. But nonetheless, we're not understanding life. We're not understanding disease without understanding proteins. And we are finally at the precipice of a new age when we can characterize proteins at a much deeper level, much more profound level that becomes commensurate with their rich dynamics and with their functions in life, making all of the reactions that keep us alive possible.